Hi anatomy students, we're now going to move on in our urinary system and look at the structure of the nephron. The nephron is the structural and functional unit of the kidney. There are about 2 million nephrons in each kidney. The nephrons will begin in the cortex and the tubules will dip down into the medulla, and then they will return to the cortex before draining into the collecting duct. As the functional unit of the kidney, the nephron is where the production of urine begins. Each nephron is made up of two regions that are connected to a shared collecting duct. These two regions are the renal corpuscle and the renal tubule. The cortex of the kidney contains the renal corpuscle, the proximal and distal convoluted tubules. The renal corpuscle is responsible for the filtration of the plasma. It contains two structures, the glomerulus and Bowman's capsule. The glomerulus is a cluster of fenestrated capillary loops that are enclosed by Bowman's capsule, which is part of the renal tubule. Bowman's capsule has two layers, a visceral layer, which is in contact with the glomerulus, and it is composed of specialized epithelial cells called podocytes, and a parietal layer, which is the outer layer, and it is composed of simple squamous epithelial cells. This layer is continuous with the epithelium of the proximal convoluted tubule. The space between the visceral and parietal layer is named Bowman's space, and this space contains the ultrafiltrate of the plasma. The other main part of the nephron is the renal tubule, which is a long tubular passageway in which renal absorption and secretion occurs during the formation of concentrated urine. It begins at the renal corpuscle and is contiguous with the proximal convoluted tubule and ends at the distal convoluted tubule. Let's look at the specifics of the glomerulus. The renal corpuscle is an approximately 200 micrometer in diameter structure that is responsible for the filtration of blood. It contains two structures, the glomerulus, which is a cluster of capillaries surrounded by a thick basement membrane and a layer of epithelial cells called podocytes, and Bowman's capsule. Bowman's capsule surrounds the glomerulus and is defined by a layer of epithelial cells that are continuous with the podocytes that surround the capillaries. The capsule itself is composed of a layer of epithelial cells, and it is connected to the initial segment of the renal tubule. It forms the outer wall of the renal corpuscle, which will be called the parietal layer, and it encapsulates the glomerular capillaries in the visceral layer. The outer layer is lined by simple squamous epithelium, whereas the visceral epithelium that covers the glomerular capillaries is covered by what are known as podocytes. The glomerulus within Bowman's capsule is a knot of fenestrated capillaries. It is covered in the visceral epithelium that's continuous with the parietal layer, and it is made up of large cells that are called podocytes. The podocytes, foot cells, are complex processes or feet, which are known as pedicels, that wrap around specialized dense layers of the glomerular capillaries. Filtration slits exist between the pedicels that wrap around the glomerular capillaries. These narrow passageways allow very small materials to pass between the filtration slits. The filtration barrier of the glomerulus is a size and charge selective barrier. The structural components of the barrier start with a fenestrated capillary endothelium with a negatively charged surface that repels many plasma proteins. Beneath the capillary epithelium is a thick basement membrane that is also negatively charged. The foot processes of the podocytes form the final barrier. Note that the slit diaphragm exists between the foot processes, or the pedicels. These play a role in filtration of plasma as mutations in genes that encode proteins of the slit diaphragms lead to proteinuria. or protein dropping out in the urine. The strong negative charge on the surface of the endothelium and within the basement membrane repel negatively charged proteins in the plasma, notably albumin. On the next slide, we'll look at these podocytes up close. And here is a beautiful electron micrograph of the glomerular capillaries. You can see an arrow pointing to the podocytes, which wrap around the capillaries. And labeled CB, you can see the cell body of a podocyte, and labeled P, you can see the pedicel or the foot process. In B, we also see the vantage point from within inside of the capillary, that you can see the fenestrations that perforate the endothelial cells. So you get a very good view of the filtration barrier in the capillary. And really know this, because filtration of blood is where the action takes place in the kidney. Here is another drawing that shows that same filtration barrier. You can see that we've taken a section out of the glomerular capsular membrane, and you can see in the photo at the bottom, or the illustration at the bottom, where you have a red blood cell that's too large to pass through the filtration slits of the endothelium. But you can see that smaller substances can come out of the plasma and go through those fenestrations. Then they must go through that basement membrane that has a negative charge on it, and then pass through the slit diaphragms in the layers of the podocytes that are surrounding the capillary, at which point they would drop out into Bowman's capsule, whatever's left over, whatever filtrates out. And here is another electron micrograph of that same capsular membrane, showing the filtration slits in the capillary endothelium, and then the layer of the dense basement membrane, and then the slit diaphragms in the visceral wall of the glomerular capsule. Mesangial cells are also found in the glomerulus, and they secrete the proteins and glycoproteins that compose the matrix that supports the other cells in the glomerulus. They are located at the vascular pole of the nephron. And just to review, because we can't review it enough, the layers of the glomerular filtration membrane include a capillary layer, which is the fenestrations in the glomerular endothelium. And then there is the negatively charged dense layer, layer of the basement membrane that is in between the capillary layer and the visceral epithelium, which forms the third layer. And significantly, the visceral epithelium is the podocyte layer that has the filtration slits in between the pedicels or foot processes. And once again, to look at that up close, you can see the red blood cell lying within the capillary, and it of course is too large to come out through the filtration slits or the fenestrations in the capillary endothelium. However, smaller substances can go through that, and they will need to make it through that negatively charged layer of the basement membrane, and then through the filtration slits that run between the pedicels of the podocytes. So our glomerulus, each glomerulus will consist of 50 intertwining capillaries. The blood is going to be delivered into the glomerulus through the afferent arteriole. The blood will leave the glomerulus through the efferent arteriole. And then it will flow into the peritubular capillaries, which will drain into small venules, and that will then return the blood to the venous system, ultimately out of the kidney through the renal vein and back into the inferior vena cava. However, in between that, coming in through the afferent arteriole, for lack of a better term, let's call it dirty blood, is flowing in, and you see that coming in through the afferent arteriole up in the corner with the red arrow. And that blood will need to be filtered or cleaned out. It's going to be the pressure of the blood, the hydrostatic pressure, or the blood pressure, that will force that fluid through all those capillaries of the renal corpuscle. And when we're forcing that blood through there, different dissolved solutes will fall out into the capsular space or Bowman's space. It will go through that filtration barrier, and that should produce a protein-free filtrate that's similar to blood plasma dropping out into Bowman's space. And the cleaned blood, from which all those solutes were removed, will exit where you see the blue arrow out of the efferent arteriole. And that filtrate is where the green arrow is, dropping into the Bowman's space, which is between the visceral podocyte layer and the parietal layer of Bowman's capsule. And at the beginning then, it will then go on into the proximal convoluted tubule. Let's look here. You can also see another slide that shows the blood coming in through the afferent arteriole off of a cortical radiate artery, or our interlobular arteries, as we also know them. And you see at number 1 is where we have the filtrate depositing in Bowman's space. And then you can see how the cleaned blood will exit out of the efferent arteriole, and this blood will travel then through the peritubular capillaries and go on to the cortical radiate veins, or what is also known as the interlobular veins. The filtrate from number 1 will then pass down into number 2 and 3, which is part of our proximal convoluted tubule. And we'll learn later on about how through the proximal convoluted tubule, you'll see at number 2 that we'll have reabsorption of some materials, and we'll also have at number 3 tubular secretion of other materials. So the main parts of our nephron were the renal corpuscle that we talked about and the renal tubule. And our renal tubule is where we're picking up now. It's that long tubular passageway that begins at the renal corpuscle. And it starts with the proximal convoluted tubule. And the proximal convoluted tubule will then join up with the loop of Henle. The loop of Henle has a descending limb, and then it makes a U-turn and has an ascending limb. And the ascending limb will then join up with the distal convoluted tubule, which will then empty into a collecting duct. So let's look at all these component parts of our renal tubule. Our renal tubule is composed largely of a simple cuboidal and simple squamous epithelium. The epithelial cells each have a primary cilium on there. So let's talk first about the first segment, which is the proximal convoluted tubule, abbreviated the PCT. The proximal convoluted tubule is the first and longest segment of the renal tubule, and it begins at the urinary pole of the glomerulus. There is a vascular pole and a urinary pole at each glomerulus. The proximal convoluted tubule is the site where the majority, about 65% or so, of ions and water in the urinary space is reabsorbed back into the body. The cells of the PCT have an apical brush border to increase their surface area. The convoluted portion of the tubule leads straight into a segment that descends into the medulla within a medullary ray and becomes the loop of Henle. The loop of Henle, also known as the nephron loop, forms a hairpin structure that dips down into the medulla. It contains 4 segments: the thick descending limb, which extends from the proximal convoluted tubule, followed by the thin descending limb. This joins to the thin ascending limb, which then joins into the thick ascending limb. The turn of the loop of Henle usually occurs in the thin segment within the medulla, and the tubule then ascends toward the cortex parallel to the descending limb. The end of the loop of Henle becomes the distal convoluted tubule near its origin at the glomerulus. The loops of Henle will run in parallel to capillary loops that are known as the vasa recta, and this will become significant as we delve more into the physiology of the creation of a concentrated urine. The distal convoluted tubule follows after the loop of Henle. Further reabsorption and secretion of ions occur in this segment. The initial segment of the distal convoluted tubule lies right next to the glomerulus and forms what's called the juxtaglomerular apparatus. Distal convoluted tubule cells lack a brush border on their apical surface. The juxtaglomerular apparatus is a specialized structure formed by the distal convoluted tubule and the glomerular afferent arteriole near the vascular pole of the glomerulus. Its main functions are to regulate blood pressure and the filtration rate of the glomerulus. The complex secretes renin, which regulates systemic blood pressure via the renin-angiotensin-aldosterone system, which we will discuss further later on. The complex also regulates pressure in the afferent arteriole in response to changes in the rate of filtration. It is composed of the following cells: the macula densa, which is a collection of specialized epithelial cells of the distal convoluted tubule. These cells are enlarged as compared to surrounding tubular cells. The cells of the macula densa sense sodium chloride concentration in the tubule, which is an indicator of the glomerular filtration rate in the nephron. In response to elevated sodium, the macula densa cells trigger contraction of the afferent arteriole, reducing flow of blood to the glomerulus and reducing the glomerular filtration rate. The JG cells, or juxtaglomerular cells, are part of the afferent arterioles, which are responsible for secreting renin. Renin increases blood pressure via the RAS, renin-angiotensin-aldosterone system. Juxtaglomerular cells are derived from smooth muscle cells of the afferent arterioles. The terminal portion of the distal tubule empties through collecting tubules into a straight collecting duct. The collecting duct system is under the control of antidiuretic hormone, ADH, which you're already familiar with. When ADH is present, the collecting ducts become permeable to water. Thus, they assist in the reabsorption of water, or antidiuresis. The collecting duct will receive the renal tubules of several nephrons, and the collecting duct of one renal pyramid converges to form one tube that opens at the renal papilla into a minor calyx. The function is generally for the collecting duct to collect the urine from the nephrons and move it into the renal pelvis and ureters. Here we see the renal papilla that is full of all those collecting ducts that are converging to have the urine collect and drain into the minor calyx. Let's now go to some practice questions about the nephron. The innermost layer of the glomerular capsule is the visceral layer, which is made up of which of the following: mesangial cells, juxtaglomerular cells, podocytes, macula densa cells, or the simple squamous epithelial cells. And the answer, of course, is that the podocytes make up that visceral layer that covers over the capillaries in the glomerulus. These structures empty urine into the renal pelvis. And these would be the collecting ducts that we just examined at the end of this lecture. Which anatomical portion of the nephron connects Bowman's capsule to the loop of Henle? The collecting duct, the PCT, the DC2, medullary ray, or ureter? And it is the proximal convoluted tubule that is in between the Bowman's capsule and the loop of Henle. Now here's one where you have to come up with the original layers yourself. So place in order from the inside of the glomerular capillary the 3 layers that form the filtration barrier in the glomerulus. So think about that, maybe pause for a minute, and we'll come to the answer. And from the inside of the glomerulus, you can see first we will encounter the fenestrated endothelium of the glomerular capillary. Next, we'll have that negatively charged dense layer of the basement membrane. And then we'll have the filtration slits that run between the pedicels or the foot processes of the podocytes that form the visceral epithelium which overlies the glomerular capillaries in Bowman's capsule. And you really need to know this. The first part of the renal tubule is called which one of the following? The DCT, the PCT, the loop of Henle, the collecting duct, or the glomerulus? And this again is the proximal convoluted tubule. The U-shaped structure in the renal tubule is known as and it's of course the loop of Henle or the nephron loop. Collectively, the materials eliminated from the glomerulus into Bowman's space is or are what? Electrolytes, urine, renin, or filtrate. And it's really between 2, and it's just to refine your knowledge here. And it is the filtrate. It is not yet urine, okay? It's urine when it makes the point of being eliminated through the collecting ducts. At this point, it is simply filtrate. We haven't gotten to forming that yet, but we did go over this part that it's the filtrate that is in Bowman's space. And so our next lecture, we're going to go over the formation of urine, of which forming the filtrate is the initial part. The kidneys control the proper balance of hydrogen ions in the blood, thus helping to regulate what levels in the body? Well, this is obvious that you know from anything involving the hydrogen ions, it will be regulating the pH. Okay? And we will see you next time. Oh, by the way, obviously the due date that I have, which is tomorrow, the 28th, is a little bit too soon. I think I was getting a little bit too hopeful as to how quickly we could proceed. About half of you still haven't turned in your digestive module questions. Okay, so I left a lot of time in at the end of the semester because I didn't know how quickly we would all be progressing. Okay, hope you're all doing well and staying healthy. See you next time.